That was a really cool introduction. I'm excited to kick off this year's minimal cell workshop by not even talking about the minimal cell. I'm Daniel from Dr. Bogomil Karas's lab at Western University, and I will be talking about some of the technologies that have enabled the creation of the synthetic cell, namely the transplantation and cloning of whole bacterial genomes, but for the species Acholeplasma laidlawii. There's a growing list of bacterial genomes that can be cloned inside of yeast as centromeric plasmids. And there's a smaller list of bacterial species that can have their genomes transplanted into Mycoplasma capricolum through genome transplantation. Currently, genome transplantation is limited to a single class of bacteria called mollicutes. And within the mollicute class, uh, genome transplantation is currently restricted to a single phylogenetic group consisting of species such as mycoplasmas and mesoplasmas. To date, all of these transplantable species use a non-standard genetic code and also require some sort of serum like FBS for culturing. This is part of our motivation for wanting to work with laidlawii. Acholeplasma is unique in that it is a mollicute species that uses a standard genetic code, and it has the potential to be much cheaper to grow because when we grow laidlawii, we don't have to add any kind of serum or any fatty acid supplementation. Another motivation we have for working with laidlawii is the close phylogenetic relationship it has to another group of mollicutes called the phytoplasmas. Phytoplasmas are plant pathogens that cause some issues in agriculture, but currently it is technically difficult to grow them in a lab, and therefore they remain understudied. So if we could get genome cloning and transplantation done for Acholeplasma, we may be able to do some creation of hybrid Acholeplasma phytoplasma strains in yeast, transplant those back into Acholeplasma, and then hopefully have a strain that would be easier to work with in a lab setting. Towards genome transplantation in laidlawii, we have selected two strains, one as a donor and one as a recipient. The donor we've chosen is strain PG8A. Bogomil has previously cloned the genome of this strain in yeast, so we know that it's possible and that presents a way to modify the strain in the future. However, in our experience, this strain seems very resistant to transformation, and for that reason, we've chosen a different strain, 8195, as a potential recipient. Strain 8195 can be transformed, and it's also thought to be deficient in both DNA recombination and restriction, which already make it a pretty good potential recipient. And there has been some genetic tools developed for strain 8195 over the past. However, at around 1995, there's a pretty big gap in the literature. And since then, it's been becoming harder to reestablish or replicate some of the things that have been done with this strain in the past. And therefore, when I first started working with laidlawii, the first few years of my research were spent attempting to recreate, reestablish, and expand on the laidlawii genetic toolbox. So I'd like to discuss some of the things that I have done for this strain to date. The first thing I did was reestablish transformation to laidlawii using a new electroporation protocol that I developed. And I used this protocol to deliver two plasmids. One of them I constructed called PAL1, which just has the Acholeplasma aureus, and the other is a plasmid called PNZ18, which has been used previously to transform laidlawii. It seems that PNZ18 undergoes large rearrangements when we try and clone sequences into it. But since then, I have been able to find a mutation in one of the replication proteins, RepB, which appears to stabilize the plasmid and allows us to use it for cloning of genes. And although we did get some decent transformation efficiencies when first starting out with Acholeplasma, if we deliver If we can establish a higher transformation efficiency, then we may be able to deliver more complex or larger constructs in the future. And towards this goal, I subjected wild type 8195 to an evolution approach where I transformed and cured the plasmid several times. And I then took this final pool of strains and screened a few for improved transformation. And we were happy to see that one of these strains showed a drastically improved transformation efficiency relative to wild type, and we've called this strain DNE, which has been deposited at the ATCC. So moving forward, I may refer to strain 8195 as DNE, since this is the strain that we typically work with now. Just know that it's part of the 8195 lineage. And at some point, I've been able to go back to strain PG8A and establish transformation using the TN5 transposase. It's still very resistant to transformation. I was only able to get one colony, but I was able to integrate the tetracycline marker TET-M into the genome, and this presents us a potential donor strain that we could use for future genome transplantation experiments. Furthermore, because there's a lot of literature on strain PG8A published, I've been able to go back to some of the transcriptional start site data available for this strain and use that to find putative promoters that we can use to express exogenous genes. And in this case, we have some nice blue Acholeplasma from laxi. So those are some of the tools that I've been working on over the past few years, and I wanted to show a new chapter of my research that's been unfolding recently, which is the use of some of these genetic tools to recreate a strain of yeast that has the cloned laidlawii genome. When Bogomil first cloned the PG8A genome at the JCVI, he found that there was actually one gene in the PG8A genome that was toxic to yeast, and the gene had to be removed before genome cloning was successful. And we know that this gene is also present in strain 8195, therefore it seems likely that we would have to remove it. And one way that we could remove it, or at least disrupt it, is through the integration of yeast elements directly into that gene to disrupt it, since these elements need to be integrated into the genome for cloning to be successful anyway. Typically, we would not be able to do this with strain DNE, as strain 8195 has a mutation in REC-A that renders it deficient in DNA repair. However, I found that I could actually restore the ability of this strain to do homologous recombination by taking the functional copy of REC-A gene from PG8A, cloning it on PNZ18, and using that to complement the strain. I've then been able to transform this complemented strain with a plasmid that has a region of homology for the toxic gene. And I'm showing here a PCR showing that we've been able to integrate a whole plasmid containing yeast elements, Senar's His, directly into that gene. So at this point, we hopefully had a strain that would be able to be cloned inside of yeast. And towards that goal of cloning the genome, I transferred the laidlawii genome to yeast through what I think is a pretty cool method called cell fusion. In this method, you don't have to isolate whole bacterial genomes for transformation. You can just transform the entire live cell into yeast spiroplasts, which causes engulfment of the cell by the yeast. And at some point, the bacteria will die, allowing the genome to be taken up by the yeast, where we can then select for it using -His. Following cell fusion, I took a few yeast transformants and screened them for the presence of the laidlawii genome. So here I'm showing a higher resolution screen that was done after a preliminary screen for a couple of strains. And this screen involves running PCR for 5 different loci throughout the 1.5 megabase genome. And I'm very happy to see that we have a few strains that seem to have the entire genome present, at least based off of this screen. but in the future, we'd like to do some sequencing or running a pulse field electrophoresis gel to further confirm that. And what I think is really interesting in addition to that is I had a second control strain where the yeast elements were integrated into the genome elsewhere, so not in the toxic gene, and the toxic gene was left intact. And for cell fusion with this strain, we see that the genome was only partially cloned. So it seems that there may have been some rearrangements that happened because the toxic gene was not interrupted. So I would like to give you a current look at the laidlawii genetic toolbox. This is not just work done by myself. This is a culmination of work that's done by members of our Acholeplasma working group. So there's a few of us that will meet every few months or so to discuss some of our most recent research with laidlawii, resulting in this current toolbox that you see. And what I think is really cool is that most of these genetic tools did not exist 4 or 5 years ago. So compared to what you can do then versus what you can do now, it feels as if the Acholeplasma field is kind of exploding, which is very exciting to see. So now that I've been able to clone the genome in yeast and kind of recreate that yeast strain, the only thing standing in the way between us and creating synthetic Acholeplasma cells is genome transplantation. We currently have a working PEG protocol that will likely need to be optimized in order for transplantation to be successful. So that's currently where I am, trying to optimize PEG transformation. But even before that, being able to restore recombination in strain D and E will allow me to start working with integrating even small or medium-sized phytoplasma pathways into laidlawii, which could be pretty interesting before we get to the point of transplantation, hopefully. My lab is also interested in the creation of designer endosymbionts, so turning bacteria into designer organelles. And just knowing how well Acholeplasma fuses into yeast, this could be another potential avenue of research that happens with laidlawii in the future. With that, I would like to thank some members of the Acholeplasma working group. I feel as if the exchange of resources and protocols between us has really enabled the Acholeplasma genetic engineering field to expand. And if you're interested in joining our Acholiverse, we are currently looking for opportunities to collaborate on grants. So if that's something that you're interested in, feel free to send Bogomil an email. And more generally, if you're interested in working with Acholeplasma and you'd like to receive some strains or some plasmids, we would be happy to send those to you. So again, feel free to send Bogomil an email. And thank you very much for listening.
Nice job, Daniel. I'm going to start questions. So in our experience, when we were trying to transplant Mycoplasma genitalium in one strain of Mycoplasma genitalium into another strain of Mycoplasma genitalium, we found that eventually we realized that the transplantation protocol, the treatment of the recipient cell with calcium, activated nucleases on the cell surface. And we did experiments where we showed that if you put PUC19 in the extracellular, in the medium around or in sucrose buffered hepes around Mycoplasma genitalium, that you would degrade the plasmid if you treated with calcium. We were, I'll say, so smart that we chose, although really we knew nothing about this, I think, that Mycoplasma capricola and mycoides, the way that we do transplantation now, they do not have this cell surface associated nuclease, unlike almost all bacteria. Have you thought about that for your Acholeplasma?
Yeah, we have seen some pretty strong nuclease activities, so we have to think about how we work with that moving forward. Something that I think is interesting is that the gene that I had to disrupt in order to make cloning of the genome possible is actually predicted to be an extracellular nuclease. So this could already be one gene that hopefully having knocked out may reduce the activity of those nucleases. Um, we have done some development, or we're starting some development of um, CRISPR editing systems to potentially knock out some genes. So that might be some way that we could tone down those nucleases in the future.
We didn't get so far as to actually try this with transplantation, or at least get it successful with transplantation. But we did show that if you did a 30-minute treatment with trypsin, the same concentration of the trypsin that we buy for stripping mammalian cells off of flasks, that will transiently eliminate the nuclease activity. So it's a thing to think about.
Yeah, we'll definitely have to try that. Thank you for the suggestion.
And then you're also going to be risking false positives because if there is homologous recombination in your recipient cell, you will get false positives where you're just bringing in your antibiotic resistance marker, because we saw that.
Mhm. Right. Luckily, we're not too worried about that, seeing as it seems that 8195 without RECA complementation is pretty bad at recombination.
Mhm.
Like if we leave the OREC plasmid for maybe 2 or 3 passes with selection, then we do start to see a little bit of integration, but only because it's able to replicate for so long. But having non-replicative plasmids with regions of homology, we've never seen integration of those without
Okay.
RECA.
That's really useful for you. That'll increase your chances.